transmembrane and secreted forms of immunoglobulin are generated from alternative heavy chain transcripts. Immunoglobulins of all heavy chain isotypes can be produced either in secreted form or as a membrane-bound receptor. All B cells initially express the transmembrane form of immunoglobulin M. After antigen stimulation, some of their progeny differentiate into plasma cells producing the secreted form of immunoglobulin M, whereas others undergo isotype switching to express transmembrane immunoglobulins of a different isotype before switching to the production of secreted antibody of the new isotype. The membrane forms of all isotypes are monomers comprised of 2 heavy and 2 light chains. Immunoglobulin M and immunoglobulin A polymerize only when they are secreted. In its membrane-bound form, the immunoglobulin heavy chain has a hydrophobic transmembrane domain of about 25 amino acid residues at the carboxy terminus, which anchors it to the surface of the B lymphocytes. This transmembrane domain is absent from the secreted form, whose carboxy terminus is a hydrophilic secretory tail. The 2 different carboxy termini of the transmembrane and secreted forms of immunoglobulin heavy chains are encoded in separate exons, and production of the 2 forms is achieved by alternative RNA processing. The last 2 exons of each CH gene contain the sequences encoding the secreted and the transmembrane regions respectively. If the primary transcript is cleaved and polyadenylated at a site downstream of these exons, the sequence encoding the carboxy terminus of the secreted form is removed by splicing, and the cell surface form of immunoglobulin is produced. Alternatively, if the primary transcript is cleaved at the polyadenylation site located before the last 2 exons, only the secreted molecule can be produced. Although the production of membrane-bound and secreted versions of the heavy chain is achieved by similar mechanisms to those that allow the co-expression of surface immunoglobulin M and immunoglobulin D, these 2 instances of alternative RNA processing act at different stages in the life of the B cell and on different primary transcripts. B cells make a long heavy chain transcript that can be processed to give either transmembrane immunoglobulin M or immunoglobulin D before they are stimulated by antigen. A B cell that is activated ceases to co-express immunoglobulin D with immunoglobulin M, either because mu or delta sequences have been removed as a consequence of an isotype switch, or in immunoglobulin M secreting plasma cells because transcription from the VH promoter no longer extends through the C delta exons. And that's transmembrane and secreted forms of immunoglobulin are generated from alternative heavy chain transcripts. Have a great day.